Thank you. Good morning, everyone. I would like to start, I think it's a must, congratulating Professor Salvatore for this event, but more important for what he has done, organizing the center change. So I think this is something that all the scientific community is happy and uses a lot, so it's really a center for us of reference. I'm also happy to have this occasion to present our interests and our work. So I will give you a story centered on membrane traffic. And as you might know, membrane traffic is central in cell function, and può far partire il movie. And it's involved in a number of processes like secretion, endocytosis, organelle division. And here in this movie, you can see how dynamic the process is. Since you already started, now you see a protein concentrated in the Golgi complex and then going toward the plasma membrane. And here you see a nice picture of the EM picture of the Golgi complex that is in fact the platform that is involved in many, if not all, of this membrane traffic steps. Now, membrane traffic is important not only because of its physiological relevance, and this is so it's not only a curiosity-driven interest that brings people to study traffic, but there are a number of monogenic diseases, and now also cancer has been clearly related to the dynamic of the Golgi complex and membrane traffic more in general. So when one studies this basic event, of course, as always, a look at what is the disease involved. Our interest in the Golgi, in the membrane traffic field started with the mechanism that lead carrier to be formed and secrete toward the plasma membrane. And in particular, my lab became interested in this field when in the lab of Alberto Luini, they discovered that not only vesicles are responsible of traffic toward the plasma membrane, but also this peculiar, let's call it, tubular carrier of, that are pleomorphic. And you can see it here in this movie, a very nice live formation of a carrier. Now, in order to be formed, any kind of carrier, they need to undergo a series of processes that start, of course, from the membrane. So whatever the membrane is, but we are talking now about the Golgi complex, what you need to do is to deform, so to bend the membrane, and then the membrane has to elongate to form the carrier that then eventually detaches with the fission process. So we have been interested in this event, namely the formation and the regulation of the formation of the fission, of the transport carrier, and in particular of the tubular carrier formed at the Golgi complex. Now, if you have to undergo such a study, how do you start? Quite a few years ago, I should say, we decided to start with the toxic process, which is quite usual in cell biology, to study a toxin that usually have as target very relevant processes in the cell, and by understanding the mechanism of action of the toxin, understand some relevant event in the cell. And indeed, we studied the Brefeldin A, which is a fungal toxin that disassembled the Golgi. So here you see a nice Golgi, and here you see what Brefeldin can do to the membrane, a totally disassembled system. Now, to make a very long story short, I mean, this has been done several years ago, what we could show is that Brefeldin A can induce the ADP ribosylation of a very specific substrate. So we thought that this substrate could be relevant in the whole process, and in this process of membrane reorganization or disorganization, let's say. We called this protein P50, and at the time we decided to purify it, clone it, and so a lot of work in one word, And we call this protein Brefeldinae adipirybosylated substrate, BARS, so I will call it BARS for all my talk. And eventually we also crystallized the protein together with Martino Bolognesi. And here you see the structure that is quite relevant for what I will say later, since it is organized like the dehydrogenases with two main portions and a Rosman fold, which is relevant for the binding of cofactors that determine eventually the function of this protein. When we cloned the protein, we soon realized that the protein was highly homologous to a protein that was cloned shortly before us, and that was called CTBP1, C-terminus binding protein 1. Now, this is a family of protein, and the interest in this family is that CTBPs are known as co-repressor of transcription, so they act in the nucleus, and thus we were facing a family of protein with a dual function. So we have that BARS can be in the cytosol or in the nucleus, and this shuttling is determined by several events, for example, the cofactor that make a monomer versus a dimeric organization. And according to the localization of the protein can serve different cell function, as I said. So this is part of the families of protein that are called, as I said, the dual function or moonlighting protein. We were interested, of course, in the cytosolic function of BARS, and we could soon show that indeed BARS was related to the protein, to the membrane rearrangement. And in vitro study, we could show that isolated Golgi membrane could be fissioned, disassembled in the presence of BARS, and that BARS indeed was part of the fissioning process that occurred at this level of the membrane. All this, of course, is in vitro, but in several studies, oh, I put it in the wrong position, sorry. In several studies that I don't have time to summarize, we could show, and this should start now, but it doesn't, yes. We could show that indeed this fissioning event is relevant in a number of cellular processes. So in the transport from the Golgi to the plasma membrane, for example. And here we also identified a phenotype that has been very useful in these years for us and for other labs to identify a fissioning defecting cell. So when BARS is not working by knocking down or the use of antibodies, you can see the formation of these long tubules that are diagnostic of a fission that is impaired. Okay, BARS is involved also in the COP1 formation, COP1 vesicle formation, and also in macropinocytosis. Here you can see how nicely macropinocytosis occurs, and it's totally prevented by, by the knocking down of BARS. So, in addition, BARS is important in the fragmentation of the Golgi during the cell cycle. Again, I don't have time to go through the data, but this suffice it to say that BARS is essential to have the first fragmentation that occurs in the cell cycle, what is called the Golgi checkpoint, which makes the ribbon of the Golgi that you see here organized in mini stacks that you see here, and then eventually the Golgi will be totally fragmented, and this is a checkpoint to enter mitosis. So I think, okay. Knowing all this and knowing that the protein is relevant in the cellular process, of course, we addressed in the last few years two relevant questions, which are, how is BARS working? I mean, what is the machinery leading to fission? And since this was our starting point, how is the ADP ribosylation involved in this event? What is the Brefeldinae mechanism of action? So to address the first point, we identified the interactors of BARS in the process. And we did this by using an immunoprecipitation approach followed by proteomic approach. And here, again, I will summarize a long series of experiments, which I'll be ready to discuss if you are interested later. Sorry. What we discovered was a number of interactors, and to our I should say satisfaction, we discovered that among the interactors there were a number of proteins already known to be relevant in the tubulation process. As I said before, in order to have carrier formation, you need the membrane to tubulate and then eventually be fission, detach in the carrier. So this protein, like ARF, FAP, PKD, were already known to be essential for PI4 kinase, of course, for the tubulation. So we concentrated on proteins that were not yet in the picture, and we concentrated on 1433 gamma. Why? Because 1433 gamma is a family of proteins, but this isoform in particular is localized on the Golgi and is present as all the other 1433 as a dimer. 1433s are known to be adaptors proteins, so our guess was that also at the Golgi level, this protein could play the adapter function. And this indeed what we discovered. Basically, what is formed at the in specific site of the Golgi membrane, of the Golgi complex, is a complex, is a tripartite complex formed by a 1433 gamma dimer that will bind PI4 kinase and bars. In order to have the transition from this situation to this situation, you need to regulate these two proteins. So these two proteins are phosphorylated by PKD and PAC, and after phosphorylation, they are recognized by 1433 gamma, and they form a tripartite complex, which is necessary to have fission. How do we know this? With a number of experiments, which again, I don't have time to show, but at least I will show you what I said before. No, I need this movie. Can you start? Ecco questo. I show you here that, again, when we impaired 1433 gamma by downregulation, what we could see was that the carrier formation was diminished, and you have this formation of long tubules that, again, are diagnostic of a system that is not fissioning, I mean, of hampered fission. And the same, of course, if you downregulate bars or if you use a dominant negative form of bars. Now, how is fissioning occurring? It's clear that this complex, this protein complex, this tripartite complex is essential for fission, but what is the mechanism of fission? Now, there are a number of examples in the literature. The best known, if you like, in membrane transport is dynamin, or the first one discovered. And dynamin is a mechanoenzyme that basically organizes a collar, let's say, along the membrane, and it reduces its dimensions so to lead it to fission. Another well-described mechanism is the one obtained by the bar domains. So proteins like epsin or SAR1 that have the bar domain in their structure, they can insert in the lipid bilayer and force the curvature so that it will be unavailable, let's say, to be in a bilayer, and they will cause the fission. Now, the third mechanism that is discussed and accepted is the lipid phase separation. Namely, in the cells, you can have domain, in the membrane, sorry, you can have domain in which specific lipids are concentrated, and this will cause a kind of a phase transition that will lead the fission and the separation of the membrane. So what was the case of our machine? And this is the work, molecular machine, and this is the work that we have done more recently, so it's not it's submitted, not published yet. We concentrated, we knew that the first two mechanisms were not occurring in our case, and we concentrated on the contribution that the lipid metabolism, the lipid metabolizing enzymes could have in this process, in the bars-induced fissioning process. why we did that? The literature has already a number of reports saying that the lipid metabolism is somehow involved in the tubulation and eventually fission of carriers. But in particular, in our lab, we could show at the very beginning of the story that Bars was coupled to the formation of phosphatidic acid. And in fact, the fission induced by Bars in Golgi membrane in vitro was coupled to an increase of this compound, and this lipid is known to be let's say, physiogenic lipid, that is, can favor the fission of the membrane. So could this be the molecular mechanism of Bars? To investigate this, we have looked at the enzyme that cause the formation of PA. So these are lysophosphatidic acid acyl transferase, so enzyme that by transferring a fatty acid on LPA, they form a PA, phosphatidic acid. And in the cell, again, there is a family of this enzyme. There are 7 isoforms. There are ubiquitous. They are in different cell compartments. But in particular, at the Golgi, there are 2 of these isoforms, LPAT3 and LPAT4. We concentrated on both enzymes, and, but now I will show you just the data on LPAT4. So here you can see that they both localize at the Golgi. I don't know if you can see it, but okay, it's visible from here. And most interesting, both enzymes directly interact with Bars. This was done by immunoprecipitation assay. Even more important to our story was to show that the LPAT activity is necessary for this fission step I showed you before. So again, can I have the movie here? Okay. You see here, again, that if LPAT4 is knocked down, you have the formation of this long tubule, so fission is not working. And you can see it here also by measuring the carriers that are formed. So knocking down of LPAT4 decreases the number of carriers, and this can be rescued by overexpressing again LPAT in the cell. So LPAT is needed in this formation of carrier, but more important maybe is the fact that LPAT, as I said, interacts directly with Bars, and Bars stimulates the activity of this enzyme. And indeed, if Bars is knocked down itself, the activity is decreased. So the protein and the enzyme are closely bound in their activity. So is Bars needed? Bars is certainly needed for the activation of LPAT4, that is then needed for fission. But what about the complex? So it's not only Bars that is needed, but it's the whole complex. So the idea is that we have the tripartite complex formed at a specific site of the Golgi membranes, and then Bars, once it's in the complex, activates LPAT. How do we know this? Because if we disassembled the tripartite complex, again, by knocking down LPAT, this maybe is in the next one, no. By knocking down the 1433 gamma, there is no activity. Yes, here is the experiment. Here we are looking at the phosphatidic acid production. So there is no production if you knock down the 1433. So you don't need only Bars, but you need the whole complex. So I believe that with this work that, I've summarized briefly, but it took quite some time, we have identified a molecular machine that leads to fission. So it's a fission event independent, let's say, from mechanoenzyme or from the insertion of a protein in the membrane, but is lipid dependent. It depends on the formation of PA that, as I said before, is a physiogenic lipid in that the transformation of LPA in PA cause a different geometry in the membrane that favors fission. If one of these proteins is impaired, or if the mechanism altogether is impaired, we have this phenotype that I showed you before, that is the tubulation of membrane. This is all nice and good because finally we know the mechanism of action of this protein, which we have been asked several times, but it took a long time also because, as I said, I'll add it in one word, as I said, BARS interacts with two L-path, and we have investigated both, and L-path3, which was our favored enzyme at the beginning, in fact, it's important for BARS activity, but in the retrograde traffic. So it's important in the formation of COP1 vesicle. COP1 vesicles are not formed if L-path3 is not active. But it's L-path4 that localizes at the Golgi in the right, in the trans-Golgi network and causes the fission of the tubular carrier I showed you before. Now, in the last 5 minutes, what about Brefeldin? I start all my story from Brefeldin. What do we know about Brefeldin? We could show, indeed, that Brefeldin, as I said at the very beginning, causes the ADP ribosylation of BARS, but this is an unusual reaction. We could show that this is a novel, if you want to say, ADP ribosylation reaction, which actually consists in the ribosylation of Brefeldin per se. So it's an enzymatic reaction that causes the formation of this catalyze, I should say, the formation of this product, Brefeldin A ADP ribosylated conjugate, BAC, we call it. And in fact, the isolated BAC can then bind to BARS. So it's a two-step reaction, but this is the enzymatic step. Again, a long story made very short by saying that we have identified the enzymes that are responsible for the reaction, and they are the ADP ribosyl cyclases. And in particular, we showed that CD38, that is an ectoenzyme known to form cyclic ADP ribose, is in fact the enzyme forming also BARS. So in a way similar of what it happens with cyclic ADP ribose, CD38 catalyzes in the presence of Brefeldin A the formation of BAC that then enters in the cytosol, and very specifically with high affinity ADP ribosyl binds, I should say, binds BARS. Okay, the enzyme is necessary the NAD, of course. We have identified the residue that is formed and so on. I mean, I also here I will not go into detail, but an important, information that I should give here is that if you look at the Rosman fold and how the Rosman fold accommodates the NADH, that, as I said before, is one of the cofactors essential for the function of the protein, and specifically for the nuclear function. So when BARS binds NADH, it is in the dimeric form, and it is a co-repressor of transcription, whereas I should say now, if it binds the acyl-CoA is monomeric and it works in the cytosol as a fission protein. In any case, if you look at how NAD binds in the Rosman fold, you can see how nicely BAC accommodates in the same position, and that's what we could show that it can compete very efficiently with the binding of NADH. Is all this doing something to the cellular functions of BARS? Yes, indeed, because BARS basically can be inhibited by BAC, so it can lead to the fragmentation of the Golgi, but what is important, by preventing the interaction of those partners that I showed you before regulate the complex formation that leads to fission, it inhibits the mitotic entry of the cell. So it inhibits the complex formation, and thus it blocks the mitotic entry into cells. So at this point, it was quite obvious to think that we have a novel target, a very specific target for drug development. In other words, the Rosman fold of BARS. And we have specific binders like BAC, so Brefeldin A derivatives. So we are now working in two directions. We have performed, sorry. We have performed virtual dockings experiment with several libraries, so we have analyzed many compounds, and we came out with 300 compounds that can compete, and we have validated the most interesting. But by most interesting, I mean those, sorry, those that are already accepted as drugs. And among of this, we have a few very interesting candidates. You see only the number here, and I cannot say which one. But I just want to show you what they do to the cell. So when I say validate, what I mean, we did the competition, experiment of competition with an ADH on bars, and then we look at the morphology of the Golgi. And here you have one of the extreme, I would say, compound. Compound 25, for example, makes bars be only in the cytosol. It completely localized bars in the cytosol. You don't have it in the nucleus. And you see that the result is a fragmentation of the Golgi complex. Of course, we have many situations in between or reverse, and we are looking for those compounds that can block both activity, because this would lead really to a good lead for cancer treatment. At the same time, we are synthesizing, and I don't have the data yet for that, back analogs that can be specific for the reaction that is, let's say, CD38 mediated, and thus can be used again as a blocker of the protein. So here we are. So we started long ago with the toxin, and hopefully we will end out with some good candidate for cancer treatment. Now, to summarize, I think I'm in time. To summarize, so I showed that we have identified a protein that is the core of a fission complex, a fission machine that includes several components, and that causes the fission of membrane at the Golgi level by activating an enzymatic activity and modifying the lipid composition of the membrane. Sorry. I showed that this process can be inhibited by the toxic process, by Brefeldin A, but this ADP ribosylation can be exploited for identifying molecules that can inhibit this process. And we have a patent that has already passed the first series of examination, and so we are working actively on this aspect. So that's why I said in my title that you can really start from the unknown and reach some, I hope, real translational science. Now, the most important slide of all is the one showing the people that did the work. Actually, the list is very long because this work has started about 20 years ago, so the list is really very long. But for the last part, let's say, Carmen Valente has been really instrumental in a lot of the data I've shown you. Antonino has done all the work on the ADP ribosylation together with Giovanna. Gabriele is the fantastic electron microscopist that works with us. And then all the other people that you see have contributed in different way to this work. Alessandro in Alberto's lab did the work together with Carmen on the lipid metabolism. Roman, throughout the years, has collaborated on the electron microscopy. And this has been a collaboration since the beginning with Alberto's lab that brought me from ADP ribosylation to membrane traffic. So we are collaborating on this. I should mention the funds that we get from different sources, and I always like to cite IRC first, that has always financed my work. And the last, the collaboration. I put the collaboration really with the chronological order. So we started with Martino, but we are still collaborating with him. Now we are crystallizing the protein with the different compound, and so on. Victor Su, for the retrograde transport. With Martin Crossley, we work on the transcription. With Mario Macchi, we had some experiment for the macropinocytosis and so on. You see also Italian. And I should say that we have started the in vivo experiment, which I didn't mention, and these are done in collaboration with Arturo Brunetti at Ceinge and at IBP. So I should say that at the end we are working also with Ceinge. Thank you so much. And I'm ready for questions.
Any questions?
Paolo.
But do you believe that bars is involved in membrane bending or in the actual membrane fission?
No, the membrane bending is precedes the fission. So I think since you have tubulation before having fission, I think that there is dislocalization of the complex on membranes that are already tubulating. And this is all the FAP machinery, let's call it, that I think is doing that.
Yeah, but still to have fission, you gotta constrict somehow the neck, so you need some level of membrane bending there. Because all these lipids, they would bend the membrane, but then it's not clear how the actual fission event would follow.
So the fission event can occur either by bending, by extreme bending, or by a organization of the lipid that is not compatible with the bilayer. Both events lead to membrane fission. If you have a high concentration of a lipid that is not compatible, requires too much energy to keep the bilayer organized, what you have is membrane disruption. So this, I believe, is part of the process. So I don't think that the complex we are showing, the tripartite, is really doing constriction and bending. And we have looked a lot for that with microscopy and so on, as you can imagine. So we don't have evidence. We don't have a clear evidence for that. We have it, for example, I showed the slide, but very fastly, for the COP1 vesicle formation. That was a collaboration with Victor Su, and there you say the neck of the vesicle of the COP1, but then there you have really a cytoskeleton, let's say, a skeleton, no, that is making the vesicle, and you see that bars is really at the neck. Actually, I can go back and show it to you. So maybe in that case there is also some bending. I'm not sure. I always had in mind that the lipid have more to do with this fission than the bending. Thank you.
Okay, so we move on to the next speaker, who is Professor Lucio Luzzatto, Scientific Director of the Istituto Toscana Tumori Florence. The title of his presentation is Somatic Mutation Rate, and Cancer. Lucio?
Thank you very much, Chair, and thank you very much to Franco Salvatore. It is my turn to offer heartfelt congratulations on the 10th birthday of Ceinge in the city of Naples, which for me is very dear and full of memories. Franco Salvatore may or may not remember that the first time we met was when I was a student in biochemistry in Genova, and my chief at the time, Arturo Bonsignore, with whom I did my degree, he was a man larger than life, and when he went to a congress, he wanted to bring his associates. Actually, in that case, the congress was in Palermo, and he took a number of people, including myself. I was still a student. It is true that I had prepared most of his relazione scientifica for the Congress, and so he took me along, but he hated he didn't like air travel. And so we drove to Naples, and in Naples we met his colleagues in biochemistry, and they escorted us to Molo Beverello, from where we took the ferry to Palermo. And on the Molo Beverello is the first time I met Franco Salvatore, who was a young boy like myself. And in Naples, then later, I was honored to be the director of the Istituto di Genetica e Biofisica, and it is in Naples that my children grew up to become teenagers. And it is in Naples that I had some of my most productive scientific collaboration, particularly with Bruno Rottoli, the late Bruno Rottoli, whom I like to remember as a great colleague and one of my dearest friends. And I know that at the time of change formation, there was a very strong input from Bruno, and I wouldn't be surprised if what became a strong emphasis in change on the study of hematological disorders and of genetic disorders, what one could call its translational vocation, has been due to a very fertile interaction between Franco Salvatore and Bruno Rottoli. With that, I'm brought to, very briefly, what I'd like to do is to review something of what is the current thinking about the role of somatic mutations in cancer, and particularly on how important may be the rate of somatic mutation in the risk of cancer and in the establishment of cancer. So as for all diseases, when going back to when I was young, in the medical school, they told me there are acquired diseases and inherited diseases. Now, of course, that was a bit crude. Today, I think it would be more correct to say that we are convinced that in every disease there is a role of the environment and a role of inheritance, and that basically we need to understand where in this spectrum each disease lies. And I have depicted here some self-explanatory examples, and the question is, where does cancer lie? I've not stuck my neck out too much when I put it right in the middle, and I think that is maybe fairly realistic, because of course cancer is not one disease, but it is so many different diseases, and for some there is a very strong inherited component. For some, there is a very strong environmental component, but this would require a seminar in itself. So at one extreme is the now classical pedigree that I'm showing here. I'm showing it again because of a personal recollection. This was one of the first families we studied with Ken Offit when I set up the Human Genetics Department in Sloan Kettering. And you don't need to be either a geneticist or an oncologist to realize that in this family there is more than its fair share of cancer, particularly of breast and ovarian cancer. And the case in point was this young woman who had this terrible history, of which she was fully aware of, and who wanted to know if she also was going to end up like that. And it was just about the time when the BRCA1, BRCA2 genes had been identified, and we were very concerned about the anxiogenic effect of mutation detection in such families. We did 2 sessions of counseling before even allowing for the test to be carried out. This lady decided she did want the test. We had identified the mutation in the mother, so we know what to look for, and luckily we were able to report to her at the next session that she did not have that mutation. And it came home to me what can be the anti-anxiogenic effect of genetic testing. On the environmental side, you all know these facts. These are the officially recognized factors of so-called environment or so-called lifestyle that are important in production of cancer. I always complain to my colleagues in environmental carcinogenesis that this is all very well, but unless you rank them, if you put them all on the same footing, it doesn't make any sense. You have to rank them. And if you rank them, that is where I put my priorities. I have an institutional commitment as scientific director of the Istituto Toscano Tumori that anytime I speak in public, I have to say this, but now I won't say it again. So this cartoon by an artist has tried to put together what I just said in a kind of a fantastic way with the question of environmental pollution, which has been up in the air, if I can use a pun, for a long time. But very recently, the International Agency Against Cancer has sanctioned that environmental pollution is a cause of cancer. It's a very, very important thing to keep in mind. So clearly, very strong inherited factors, very strong environmental factors. This is a diagram of what is universally accepted as the model of cancer formation, which is very familiar to all of you. So I'm only using it for pointing out two things. One, which I find very interesting from the general biological point of view, is that we were taught genetics as the science of inheritance, but already in the 50s, Pontecorvo and others developed what was then called somatic cell genetics. And perhaps even they did not know how important that would become because cancer is a genetic disease of somatic cells. And to expand on that analogy between inherited genetics and somatic cell genetics, here we have a perfect example of how Darwinian selection, classically in biology of populations, acts at the level of the microcosm of cell populations, whereby, of course, each mutation must give a slight growth advantage in order to give an expanded clone that is a target, each cell of which is a target for the next mutation, and so on. Here I've used the word, the convenient figure n, because I don't know the number of events that are required. Professor Salvatore a few days ago asked me a very shrewd question on that, and I still don't know. This is why this population, which is one step short, I call it n-1. This is particularly important because of obvious reasons. But I will stick my neck out. Already some years ago, I suspect that I wrote somewhere that probably it was between, the n number is usually between 3 and 6. Probably it varies with different types of tumors. And very recently, there's been some confirmation of that figure. So it was already more than 20 years ago when we said leukemia is a genetic disorder of hematopoietic cells. I remember that at the time, the British Medical Journal told me, well, that sounds like a very bold statement, and I insisted upon it. Now it looks like it's commonplace. It's obvious. So what do we need to know about somatic mutations in cancer? How many? I've already stuck my neck out on that. Which cells are mutated? I'm skipping that because the next talk by Pierpaolo Di Fiore, I think will show a marvelous example of that. Unless the mutations happen in cells which have a long future, they're probably irrelevant to cancer formation. In fact, there is, I think, a very interesting concept in biological evolution that perhaps one of the selective pressure on the very development of stem cells in normal tissues may have been that if every cell was a stem cell, the risk of cancer would be incalculable and intolerable. We would all die of cancer before the age of 1. So I think that's selection pressure to have stem cells, and we will hear more about that. Now, what causes mutations? I've already dwelled on 2 important factors, environment and the inheritance, but I think that it is very important to consider that for all the mutagens and for all the various mechanisms of genomic instability, the main primary basis of mutation is that DNA replication is an incredibly faithful process, including the proofreading and the mismatch repair and all that goes with it. The rate of error is extremely low, but like for every physical apparatus, it cannot be 0. And we should not forget that the first cause of mutation, therefore, is stochastic nature. This is the please don't quote me on these figures. They may be off by an order of magnitude, but even if they are off by an order of magnitude, this is the estimated rate of somatic mutation in an individual gene. This is the approximate total number of cells in an adult. By the way, some adults are bigger than others. I'm among the smaller ones. It turns out that the number of cells is about the same. The approximate number of deep cell divisions, deep having to do with the stem cell and progenitor cells, to make an adult is 10 to the 15. And the ultimate result that I like to point out is that the expected number of somatic mutations accumulated in a normal adult is of this order. Of course, unfortunately, it goes up with age. I don't like it, but it's the truth. So we are working against the background of an enormous number of somatic mutations, the majority of which, of course, are irrelevant. The majority are what in Darwinian evolutionary language we call neutral mutations. So what genes are mutated? Well, if I was to dwell on that, I would have to review all of cancer, you are all, and all things that you are familiar with. So genes involved with the cell cycle, genes involved with signal transduction, genes involved with all the basic processes, with all of the cell biology. Not only Hanahan and Weinberg, but also others have written wonderful papers on that, and I refer you to those. But lastly, I want to mention something about what kind of mutations, because we expected from the beginning, it was to be expected, that it would be the same sort of mutations that we already knew from classical inherited genetics. And of course, that is true. We have point mutations in cancer. I think it is in fact particularly interesting that some point mutations keep recurring, and when a missense mutation keeps recurring, it almost certainly means a gain of function mutation. And I have a slide which I haven't had time to include, listing those, but that list, I have to actually update that slide all the time. Only yesterday we had a seminar, very interesting seminar by Jacopo Petrini on thymic epithelial tumors, in which he has discovered a recurrent point mutation in a transcription factor. But of course, everybody knows about the myeloproliferative disorders, the famous JAK2. Everybody knows about the mutation in the famous mutation in the BRAF gene that is found in melanoma and many other tumors. Those are point mutations that are almost certainly gain of function, and of course they're very good therapeutic targets. We have an abundance of copy loss mutations and copy gain mutations. We knew about those from inherited genetics, all of those that I've mentioned until now. But when it comes to reciprocal translocations, that has been really the first major contribution of tumor genetics to general genetics, when it was discovered that if the break is across genes, then you create fusion genes. And of course, this has been the story of CML and of PML and of an increasing number. As a hematologist, I was often told in the cancer research field, oh, that is something special about hematology. You are always the odd one out. It has nothing to do with epithelial tumors. Of course, that has been amply disproven. Half of the prostate cancer have a fusion gene, and more are being discovered. In fact, in a count that was done 10 years ago, the majority of genes that had been identified as mutations in human cancer were translocations. And I think that is probably still true. And here is a whole list of fusion genes that are found in epithelial or other tumors other than hematological tumors. And each one of you may recognize something of their own interest in that list, which is, of course, incomplete. It doesn't claim to be complete. But why I'm emphasizing this is because this has been a new fact which we didn't know from inherited genetics. And more recently, there have been two rather spectacular developments. One has been this incredible phenomenon whereby a chromosome can essentially break to pieces and then be reassembled. This has been called the chromatotripsis. Interesting conjugation or combination of genetics and surgery. It's still very poorly understood how it can be that the broken chromosome finds a way to recognize the parts and reassemble, but as it were, using the Lego pieces in a different way. Very recently, another intriguing phenomenon has been discovered, namely that at the site of translocations, you're all familiar with this new representation of the human genome. I always tell the students, we've always insisted that the difference between eukaryotes and prokaryotes was that the eukaryotes have a circular genome, we have a linear chromosome, and now this representation seems to fool us. But of course, it's only a graphic gimmick to make it easy to see all these translocations and fusion genes. And the individual chromosomes remain what they were. But what is very interesting is that in the proximity of these fusion genes due to translocation, there is an accumulation of point mutations as though those have become sites of somatic hypermutation, which is reminiscent of what happens physiologically in the immunoglobulin and the T-cell receptor genes. The mechanism of this is, to me, very intriguing and not yet elucidated. Today, the model that I showed you before, I told you most of people accept it. I know there are still some skeptics that don't accept it, but in fact, it's no longer a model, it's a reality. And nowadays, using current technology, it is possible, in fact, even not too difficult, although still rather expensive, to characterize each cancer genome fully in this way, with a full inventory of the point mutations, the copy number variations, and the fusion genes. it is quite clear that the ultimate nosography of tumors will be the set of somatic mutations that are present in a particular tumor. This has also vindicated what I said before about numbers. Many people find it, understandably, all of us find it intriguing, confusing, and a challenge to bioinformatics that too many mutations are found all the time. However, let me show you just this one example, retinoblastoma and ovarian cancer. And these are just the copy number variations, but it's symbolic of most of mutations. A lot of mutations, very few mutations. Some people found this disappointing. I find it extremely gratifying. The fewer the mutations, the more you know that those are the responsible mutations for the cancer. So, as I said, the question is to identify which are the driver mutations that begin the cancer. And in the case of retinoblastoma, which is an infancy cancer, not surprisingly, this is easier today. So finally, I want to come to some work we've done recently, which has to do with the importance of the rate of somatic mutation. If somatic mutations, as I showed you, are the basis of cancer, then one item I did not touch upon is you need more than one mutation, so it's very important to know at what rate somatic mutations actually happens. Because if we simplify the production of a tumor, then it would be one mutation, then another mutation, then another mutation, and then perhaps you have a cancer cell. That is if mu is low. But what could happen is much faster if mu is higher. So can we measure mu, and what is mu in different people? First of all, there are many human diseases that are associated with genomic instability, where presumably mu is very high. Here is a list which I had all to the courtesy of Antonio Musio, who is an expert on the cohesins. And of course, if we ask this question, it has been a long debate about genetic instability in tumors, which is a very common finding in tumors, but the real question is not whether it's there. The real question is it just an accessory? Is it something after the crime has happened in an established tumor, or is it an early feature? And many people, you hear this almost as a commonplace. Oh, genomic instability, therefore he got the cancer. Well, it's a very fundamental issue. Do you need the genomic instability, or do you not? My working hypothesis has been that in most cases of cancer, of course leaving out those when there is evidence of inherited genomic instability, you don't need genomic instability to give cancer. You need those mutations. But unfortunately, because of the large number of mutations, there may not be genomic instability. And so we thought that we need to confront this quantitatively by actually measuring the rate of mutation. And we took advantage of something that emerged in the study of a rare disease called paroxysmal nocturnal hemoglobinuria, precisely the one on which Bruno and I collaborated. And some years ago, David Araten in my lab asked this question. We all know that the PNH is due to the presence of a clone that is mutated in a gene called PIG-A, which prevents the synthesis of GPI-linked proteins because it's a biosynthetic of the GPI. So we asked the question, what about normal people? And here you can see granulocytes from a normal person. At least, well, David Araten told me, you're not that normal because you've studied PNH for too many years. But then we repeated the test on many other people, and it was the same. There is a small population of cells that have the PNH phenotype in the majority of normal granulocytes. Here there were 8 per million. David was able to FACS sort these cells, and most interestingly, was able to prove that these cells he amplified the exon 2 and exon 6 from 1 to 5 granulocytes, and he found the same sort of mutations that we knew were present in the PGA gene in PNH. So we all have PGA mutations, not surprisingly. I think if you look, here we have the advantage that we have very high sensitivity to do this analysis. And in fact, what we did is we made lymphoblastoid cell lines, we grew them up. Here is from a PNH patient, here are the normal cells, and here are the PNH phenotype cells. So we sorted here from a normal person, the same thing is seen. We purify them, that is, we removed all of these, and then we grew up the culture again. Here you can see that there are none of these cells because we've removed them by sorting, but after 27 days in culture, we find them again. So I was very excited by this, that we could actually see the product of the somatic mutations that had happened under our own eyes, in our hands. And so we were able, in fact, David Iraten did very nice work on leukemia and lymphoma to ask that precise question. That is, by using this approach, can we determine the mutation rate in tumors? And here you can see a figure which we regard as rather high, and here a figure that is very high, supporting that these tumors had a high mutation rate. But here you find the famous Jurkat cells from a T-cell ALL with a essentially normal mutation rate. And in fact, by looking at several dozen cases, David showed a bimodal distribution whereby some leukemias have and lymphomas have very high mutation rate, but quite a number have mutation rate within the normal range, indicating that mutation rate increase can happen, but it is not a requirement. These were patients who had acute leukemia, some of whom died of it. They had normal mutation rate. We went further and we wanted to establish normal values for this parameter. For that, we used a surrogate, which is called the mutant frequency, which can be done directly on granulocytes. As you can see, it is a very highly reproducible technique. The intra-sample and intra-individual variation is quite small. That is, a repeat sample taken next month will give the same result. And here you can see that the distribution of the what we call the mutant frequency in log transform is a perfect Gaussian distribution. And of course, the obvious question is there a difference in the risk of cancer in those, these are all normal people, 142 normal donors. Is there a difference in the risk of cancer between this tail end of the distribution and this tail end of the distribution? That is something that we are testing at the moment. The advantage of PGA as a sentinel gene to measure somatic mutation is very summarized here and is very clear. In fact, of course, my total inability to capitalize on these things is very well known, but a company in the United States has used the PGA assay, has made it into a commercial assay, and it is now widely used in the mutagenesis field to screen for mutagens in a variety of systems. So we are very good friends, although they are making the money and we have not. I think I'm running out of time, so I will conclude by simply pointing out what are the biological correlates of mu, and by pointing out where I started from, that is the stochastic nature of the events, be it genetic or be it epigenetic. For the epigenetic, I use a freeze because it's probably freezing chromatin in a certain conformation, that are stochastic events. And therefore, what I started from is that the somatic mutations are chance events, and the chance is at the center of cancer formation. Inheritance works through chance by increasing the risk of mutations. Environment works through chance by increasing the rate of mutation. And that was very early on dictated by the myth of the blindfolded goddess Fortune. And also, as usual, science was preceded by artists. I think this phrase, the debate between Brutus and Cassius, as is it chance that determines fate, or is it ourselves that we are the underlings? And that is what we are still living with in understanding cancer. And I'd like to thank very much my small group, which is part of a much larger concern, the Istituto Toscano Tumori, which is a network bringing together all the cancer research and clinical cancer activities within the region of Tuscany, and whose mission is to understand, to treat, to prevent cancer at best for all. And I invite you all first to visit our site, www.itumori.it, and then to visit the Institute itself. You'll be very, very welcome. Thank you.
I thank you very much of the remind of Bruno's work. And yesterday we had, instead of coffee break, we had a short break made by one of the greatest Italian saxophonists who played a couple of pieces. One was in memory of just written on memory of Bruno Rotoli. He played yesterday with the saxophone that was owned by Bruno and gave to him. And the Ceinge has done, in memory of Bruno, a Ceinge Rotoli Jazz Research Ensemble. Thank you very much.
Thank you.
Our next speaker is Pierpaolo Di Fiore from Firchi Institute of Molecular Oncology in Milan. The title of Pierpaolo's presentation is The Stem Cell Compartment in the Mammary Gland, Connecting Cell Fate to Tumor Suppression. Paolo?
Thank you. Please have the slides. Always very nice to be here. We've done a lot of stuff with Ceinge in the past and presently with Franco. Ceinge is not 10 years old, actually. It's more of a concept that has been pushed through the years. I grew up with the idea of Ceinge when Franco invited me for the 10th anniversary. I said, no, you've been around much longer. But, well, what can I say? Franco, of course, is an emperor, so I will use the Chinese imperial address to wish luck to his institute, 1,000 years. Now, what I will try to do in my presentation here is to show you how in the stem cell compartment and in the subversion of the stem cell compartment that happened in cancer, there is a very intimate connection between the machinery that determines cell fate and the machinery of tumor suppression, and that actually the two machinery influence each other, and that the determination of cell fate is actually a mechanism of tumor suppression in itself. Now, in order to do that, I will have to give you a very brief introduction about the dynamics and the kinetics in the adult stem cell compartment. In normal stem cells, the characteristic of the first division of a stem cell is to be asymmetric. Now, asymmetry here is a functional concept, means that at this division, one of the two progeny remains stem, while the other one is created into something different, which we call the progenitor. As I will try to explain, this is actually due to the fact, among other things, that there is molecular machinery, proteins, that partition differently between these two cells with a variety of mechanisms, so that the two progenies are actually intrinsically different. But from the functional point of view, the relevant thing is that this stem cell withdraws immediately in quiescence, while the progenitor expands very rapidly, 8-10 rounds of duplication, but eventually all the progeny of the progenitor becomes differentiated cells. So when we look at the tissue is really made of the progeny of the progenitors, but the only way that the tissue can be regenerated is if this stem cell re-enters the cycle. Now, this from the kinetic point of view has an interesting implication, and is that the best you can do is to make one stem cell from one stem cell. So if over time, or over passages in culture, with whatever kind of assay you can imagine, I will try to show you one briefly, you count the number of stem cells in a tissue or in a model system, the best you can have is to have a flat line. Stem cells cannot increase in number. You can make one stem cell out of one stem cell. As a matter of fact, what happens is that at every passage, there is a pretty much stochastic possibility that the stem cell that we drew into quiescence undergoes apoptosis or senescence or whatever. And so actually what happens is that over time, stem cell decrease in the normal compartment, and many people believe that this is one of the reasons why we age. We run out of stem cells to repopulate our organs. Well, what happens in tumors? What we showed a few years ago in the case of the breast stem cell compartment in tumors, together with Pier Giuseppe Pellicci, is that really the characteristic of the stem cell compartment in breast tumors, but I would make a generalization because it has been actually shown in many other settings, is that stem cells oscillate between rounds of asymmetric division and rounds of symmetric division. So what does this mean? It means that sometime the stem cell will divide and give rise to a progeny which is made of two stem cells which are identical to the mother and identical to each other. In other cases, the stem cell actually replicates with an asymmetric mode, and so gives rise to a stem cell and to a progenitor, and then the tumor is actually made very similarly to a normal organ to the progeny of the progenitors, which will eventually differentiate, although with an aberrant pattern. And we proved, I will not have time to do into that, that really aggressiveness of tumors correlates with the rate of skewing of the pattern of division from asymmetric to symmetric. Or in other words, the more time that the stem cell decides to divide symmetrically, the more stem cells you will have in the tumor, the more aggressive the tumor will be. And this is summarized in the counterpart of the kinetic which I showed you before for the normal stem cell compartment. So if you count stem cell in an organ or in a model system, in vitro or ex vivo, over passages, over time, whatever, in a tumor compartment, you will have an exponential growth of the stem cell, which you can translate, in other words, by saying that the stem cell compartment has become immortal as opposed to be mortal, as we saw in the normal setting. Now, as I said, what I will try to show to you here today is the connection that in the stem cell compartment happens between the cell fate determination machinery and the tumor suppression machinery. And in order to do that, I will use the two prototypes of these functions, which are on the one hand the protein NAM, and on the other hand p53. Now, NAM is a protein which some of you or many of you might not be completely familiar with, but in the field of stem cell determination and cell fate determination, this is really a cornerstone of the entire foundation of our knowledge today. NAM is the first protein that was identified as a cell-autonomous determinant of cell fate. And this dates back now, perhaps 25 or 30 years. It was isolated in Drosophila. NAM does a very simple thing in many systems. It partitions asymmetrically at the mitosis of a cell whose progeny will undergo different fate. So in this case, NAM was isolated originally in a developmental system in Drosophila in a particular cell called the sensory organ precursor that gives rise to the bristle, which are the sensory organ of the Drosophila. It doesn't matter very much. The bottom line is that when this cell, the sensory organ precursor, divide, you see the NAM goes to one pole of the cell, and so it goes only into one of the progeny. And a lot of work has been done, of course, on this protein and on the mechanisms. I will not dwell into it, but this simple asymmetry in the two daughter cells creates a series of reinforcing circuitry that makes the two progeny to assume completely different fate. As we will see, this is very relevant also in stem cell in humans. Now, we got interested in NAM for several reasons. One is because we proved many years ago that it's an endocytic protein, and my lab has been historically interested in attenuation of signaling, but mostly because we showed that actually NAM is lost in many tumors, and in many tumors of a high relevance. NAM expression is lost in almost 30% of breast cancers, and is lost in almost 30% of lung cancer. I will concentrate on breast today. We have proven that the loss of NAM in this case is due to hyperdegradation. So the protein is normally synthesized, but the machinery that normally degrades NAM, which is a ubiquitination-based machinery, is overactive in these tumors, and therefore in 1/3 of breast cancers, NAM is lost. And we have proven that this has pathogenetical relevance, is causal to these tumors. Now, how does p53 enter the scenes? Well, it entered the scenes by a discovery which, really happened almost by chance in the lab. We were going after something else, and we ran into p53. But Ivan Kolaluka in the lab a few years ago proved that NAM, in addition to some of the canonical functions that were attributed to this protein, for example, to inhibit the Notch pathway, has a very important function in the stabilization of p53. Essentially, what NAM does is to enter in a complex with p53 and with this protein here, which is called MDM2, or sometimes HDM2 in humans, which is a ubiquitin ligase. Now, what MDM2 does as a job is to bind to p53 and to ubiquitinate p53. And the ubiquitination of p53 is the signal for p53 to go to the proteasome and therefore to degradation. When NAM is present, the activity of MDM2 is inhibited in this tricomplex, and so p53 is stabilized. So one of the simple equations which I will ask you to bear in mind to follow the rest of the talk is that high NAM means high p53, and vice versa, low NAM means low p53. Now, how do we study stem cells? I will have to give you a little brief crash course of how we purify and study stem cells, because some of the experiments I will show you heavily rely on these techniques? Well, stem cells are the only cell in an organ which can withstand anoikis. They can grow in suspension. They give rise to spheroids, which are clonal, which we call mammospheres. So a simple assay to count stem cells, it's simply to put an organ in culture and counting the number of mammospheres. And you can passage mammospheres, and so this is an assay in which you can actually keep on counting stem cells, because every mammosphere is the clonal expansion of a single cell. But then even in a mammosphere, there are 400,000 or 500,000 cells, and as predictable, there is only one stem cell, which is the daughter that withdraws into quiescence while all the progenitor proliferate. And so how do you purify it? Well, one way of purifying it is to stain the entire population before the suspension culture with a dye that goes in the membrane. We use these dyes of the family of the PKH dyes. So every cell is stained, but then when the sphere is formed, of course, the stem cell at the first division will divide the dye equally between the two daughters, but then one of the daughters, the stem, will go into quiescence, the other one will keep on dividing, so the dye keeps on diluting, and at the end you end up with an image like that in which you have a single cell. Here you can see very clearly, and you can sort, at this point, from the mammosphere, the PKH positive from the PKH negative component, and we have proven that the PKH positive component is actually a more than 90% enriched population of pure stem cells in the case of the mammary gland here. And when you have a population of pure stem cell, now you can actually follow with your eyes what a symmetric division and an asymmetric division look like. The only thing that you have to do, when I say the only thing, I pay a tribute to 2 or 3 PhD and postdocs that spends an average of 12 hours a day at the microscope to follow one by one the division of the cells, is to put them under a microscope and to do time lapse. And as you see, when these cells are red, of course, because they come from the PKH purification, they are stem cell. When you have an asymmetric division, you have the first division, but then you see that one of the daughters stays put, doesn't divide anymore. This is the cell that has remained stem, while the other divides. So in an asymmetric division, we count in a rather atypical way, we count 1, 2, 3, 5. In a symmetric division, of course, after the first division, these two cells are identical, and so they will keep on dividing the same way that we are used to look at cells dividing in vitro. So in this case, we count 1, 2, 4, 8. So when you follow these patterns, you can clearly follow a pattern of symmetric division and a pattern of asymmetric division. Okay, now to the real story that I want to tell you. How are these machineries, the machinery of cell fate and tumor suppression, connected? Well, the story starts when, in collaboration with Pier Giuseppe Pellicci, we proved that in the absence of p53, there is a dramatic skewing in the stem cell compartment from asymmetric to symmetric division. So that established the connection. Up to that point, symmetry versus asymmetry was only considered a problem of cell fate determinant. We linked it to the machinery of tumor suppression by directly implicating p53 with genetics. And so this is pretty simple. If you take a mouse, if you do, mammosphere culture, so if you count stem cell, as I told you, in the wild type, you will see that slowly they tend to die, while in the p53 minus strain, you see that they grow exponentially, so they become immortalized, and actually there is a dramatic skewing from asymmetric to symmetric division, counted with that methodology, which I showed you before. Well, what about NAM? NAM should be connected to this. After all, NAM is a cell fate determinant, and we had proven before that NAM is upstream of p53 because it should control the levels of p53. So the entire idea here is perhaps lack of NAM means low p53, as we showed in the tumors, and therefore low p53 might mean skewing of the stem cell compartment towards symmetric division, and therefore towards immortalization and cancer. But in order to prove that, we had to analyze how NAM was behaving in the stem cell compartment in the mammary gland. And fortunately enough, in this compartment, NAM behaves as it's supposed to behave, so it divides asymmetrically at the first mitosis. This is the first mitosis of a PKH positive cell, and this is an immunofluorescence for NAM. You see that NAM only goes into one of the two daughter cells, and we can prove this in different ways, but I think this image is pretty telling. And the question now is, well, NAM goes into one of the two daughter cells. Which one? The stem cell or the progenitor? Well, this can be done if you are able to isolate stem cells, as we are. We can transfect them with NAM, and now we can follow retrospectively where NAM stays during the division. So you see that during the division, this is a typical asymmetric pattern. You see there is one cell that stays put, which is the stem cell. If we follow the PKH, which here comes in a green flavor, you see that the PKH stays into one of the cells, which is actually the one that retains the stem cell fate. And if we follow NAM, you see that NAM is mostly retained in the cell, which correspond to the stem cell. So at the division of the stem cell, NAM stays in the cell that retains the stem cell fate. Well, what about P53? The idea here is that NAM should stabilize P53 in that cell, in the stem cell, and this is actually what happens. You cannot follow really P53, but you can follow the activity of P53 using a GFP hooked up to P53 responsive element. And by doing the same retrospective reconstruction which I showed you before, you see that during this asymmetric division, one cell is always stained by the PKH, which here comes in the red flavor, and in this cell, the activity of P53 is high. So at the asymmetric division of a normal stem cell, NAM goes in the cell that retains the stem cell fate, and in that cell stabilizes P53. And that's the reason why I believe that that cell withdraws into quiescence, because it has very high level of P53, while all the progenitors have lower level of P53, having lower level of NAM, and so they are allowed to proliferate. Well, this has implication. Yes, the first implication is that, to link now back to tumors, is that if this is really the mechanism through which NAM and P53 works in the stem cell compartment, then the knockout of NAM should phenocopy the knockout of P53, and this is exactly what happened. We have done it both by knocking out NAM specifically in the mammary gland. We have done it also by knocking it down in normal cells. The result is the same. You see that basically the phenotype of the NAM KO and the P53 KO is indistinguishable. This cell mostly divide symmetrically. So when you take out NAM out, now there is no P53, and so the cell behave functionally as a P53 minus strains, and there is a complete phenocopy of the phenotype. But most importantly, the idea here now is that if NAM is upstream of P53, and if loss of NAM in tumor means actually loss of P53 and therefore subversion of the stem cell compartment, you should be able to intercede with loss of NAM by restoring the level of P53. Since we are in a P53 wild-type background here, of course, we can do it with a drug which is called natalin, which is a drug that inhibits the degradation of P53 by interfering with MDM2. And this is exactly what happened. So if you take, this is done in a KD background, but the same happened in a KO background. If you take a NAM KD, you will see that the cells from the stem cells from the NAM KD expand. They are immortal. They divide symmetrically. If we treat them with increasing concentration of natalin, you see that they go to a mortal behavior, which is basically indistinguishable from the behavior of the control cells, of the wild type before the KD. And if we go and look at the symmetry versus asymmetry by doing all the little cell counting that I showed you before, you see that in the cells not treated with natalin, these are KD, NAM KD, not treated with natalin, most of the division are symmetric, as you would expect of a NAM knockout P53 null background, but when you put natalin, you recover a substantial amount of asymmetric division. So restoring P53 activity reverts the NAM effect. Well, you said this is all good in a model system, but what happens in real tumors? Well, in real tumors, exactly the same happens. Here we took a number of tumors, I'm showing you 4. 2 are NAM negative, as you see in the staining, 2 are NAM positive. The idea here, the underlying idea, which I will try to champion, is that in these tumors, the expansion of the stem cell compartment and the subversion of the compartment is due to lack of NAM and to lack of p53 and to increased level of asymmetric division. In this case, of course, I have no idea what the genetic alterations could be, but they represent our control, and we selected our control in a way to have the same tumors with the same rate of immortalization and expansion of the stem cell compartment. But remember that the underlying idea is that here the effect is due to lack of p53 due to lack of NAM, and here is due to God knows what, but it's my control. And indeed, if we take these tumors now and we treat them with Nutlin, so we treat them with a drug which is completely extraneous to the genetic alteration in these tumors, which is concerned with NAM, you see that this is the kinetic of expansion of the two NAM-low tumors. As we treat them with Nutlin, we flatten their expansion and the compartment reverts to a normal kind of behavior, while in the, sorry, there is a mistake here, these are NAM-high. I keep on loading the wrong slides. In the NAM-high tumors, Nutlin has no effect. And this can be also visualized by going and looking at the symmetry versus asymmetry. You see that in the NAM-low tumors, all the divisions are essentially symmetric, but when we put Nutlin, we recover a very significant amount of asymmetric division in both the example, while if we go in the NAM-high tumors, in the one in which allegedly the effect is not through p53, you see that the divisions are asymmetric, but Nutlin, so the restoration of p53 doesn't sort any effect. And so the idea is, what about the growth of these tumors in itself? Well, we did one last experiment here, which was to take NAM-low tumors, to let them grow to a certain size in the mouse, and then to give them a single injection of Nutlin. So this is not repeated treatment, this is a single injection of Nutlin. And then we let the tumor grow. And interestingly enough, the tumors grow at the same rate whether they were treated or not treated with Nutlin. So you might say, this is the end of the experiment. Well, actually not, because if at this stage you take these tumors and you transplant them in a second animal, now the tumors treated with Nutlin starts to grow at a much lower rate. Now, this is exactly the mechanism you would expect of a stem cell specific targeting, because here, if you are sterilizing or nearly sterilizing or quasi-sterilizing the tumor of the stem cell, you are still full of progenitors that will keep on growing at the same rate, whether there are stem cells or there are not stem cells in the tumor. So the tumors will keep on growing. But as the progenitors approach the limit of their ability to grow, the tumor can only be refueled by additional stem cell. Of course, we cannot let the tumor grow in the animal more than a certain size for ethical reason, so the only way of unmasking this is through transplantation. And you say, okay, but this is speculative. Show us the actual count of the stem cell, and this is what we did. So basically, you can take the tumors at this stage and go in serial endpoint dilution in mice, so do a titration of the amount of stem cells by doing a Poisson distribution, and you see that the treatment with Nutlin causes, in the range of 7-8-fold decrease in the number of stem cells, but not in the number of progenitors, because the tumors had expanded with the same rate. So I guess the take-home message of what I've tried to show you today is that these two proteins, NAM and p53, which have been the champions of cell fate determination and tumor suppression, are actually very intimately coupled in what we can consider the same circuitry of fate determination and tumor suppression in stem cells. They are actually both regulated through degradation event. Degradation event in tumors of NAM are, we still don't know the cause. We simply know that NAM is degraded more actively in certain tumors. We have an idea, but I will not have time to go into. P53 is degraded to MDM2, but the two circuitries are coupled because NAM stabilizes P53 by inhibiting MDM2. When the situation is skewed in this direction, we have a normal stem cell compartment in which cells divide asymmetrically. When the situation is skewed in this direction, because of lack of NAM and therefore because of low levels of P53, or because of primary alteration of P53 downstream of NAM, we have a cancer situation, a situation like this, in which the stem cells divide more frequently in a symmetric way than in an asymmetric one. But the process is reversible by what I think is a real example of stem cell target therapy in this case, by restoring levels of P53 and reconverting the skewed pattern of symmetric division towards asymmetry. And this has been the work mostly of two people in the lab, Salvatore Pece and Daniela Tosoni, during the years. Ivan Kolaluka is the guy who identified the regulation of NAM over P53. It's the result also of a long-term collaboration with Pier Giuseppe and his group, and with a number of people in the pathology department and in the various surgical department of the Istituto Europeo di Oncologia. Thank you.
Is there any question? Dr. Walsh?
A really wonderful talk. I really appreciate it. I had a question in terms of the relationship of the stem cells you see in terms of what we typically identify with mammosphere growth, like the acquisition of cell surface markers like CD44 and CD24. And the other thing is we also recognize the insensitivity to various chemotherapeutic reagents. So I was wondering, do you see that type of signature emerging when you do these knockdown approaches and knockout approaches with these cells?
Okay, well, it's a very complex question to ask, but there is an entire topic here which I didn't go into, and it's that when you knock out NAM, actually you do two things. I told you one part of the story here, which is you skew the stem cell towards symmetric division. But NAM has also another role. It induces in the progenitor epithelial to mesenchymal transition and reverts progenitor to a stem cell state. So under those conditions, we can more clearly follow some of the things that you're saying, like reacquisition, for example, of surface markers, which are characteristic of stem cells, like CD49 and the like.
Thank you very much for summarizing this beautiful story. You said in the case where you see NAM in the histochemistry, you said something like...
I have no idea what the alteration is in there.
So,
but I need a control in my experiment.
But conceptually, I think what you're saying is that there is something that overrides that mechanism.
Well, what I'm saying is that, you know, there is more than one way to skin a cat, and
yes.
The animalist will bear with me for that, and that therefore you can skew the mechanism of asymmetry versus symmetry, perhaps in different ways. In some tumors, this is due to loss of NAM. In some other tumors, it's due to I don't know what. What we were careful enough here was to choose all tumors which were P53 competent. So I know that in those cases it's not due to alteration of P53, but it might be due anything else.
So I'm saying the obvious, that to find what are the mutations in those where you see NAM and that overrides that mechanism could be very crucial.
Yes, if you are interested in what is not dependent on NAM. One has to have a hobby. I will stick to NAM.
Thank you very much.
Thank you, Mr. Chairman. Being the last speaker, I leave my acknowledgements to Franco at the end. I close the door at the end of my presentation. And in the next 20-25 minutes, I try to tell you what is the path moving from the research to the laboratory medicine for this recent technology. There are, for our patients, a very strong opportunities for improve the clinical care, but in the same time I look a lot of risk to give the wrong information, and the lab has in front a lot of challenges. This is the goal of my presentation. There is no doubt that in the last decade, more or less, there are so many new information that give us the opportunity to move from research to the clinical pattern. You see that with the last information, we are really to detect many new syndrome and to define the basic of many genetic disorders. For sure, the sequencing is not only devoted to know the human being, but we want to reach also the cell. You heard from the previous speakers that I'm a guy working in genomic, but also the somatic cell are very important to be defined also depending on the genetic background. The NGS is, there is no doubt that is a mix, for the lab is a problem, a mix of wet activity, a strong bioinformatics in which we are not skilled, and also there is all the clinical part that is not so frequently applied in many clinical centers. And there are many technologies. There are technologies that will change in a very short time. This is one of the practical problems of the laboratory medicine. Now we have already very good instrumentations, but there are new on the market. There is a long list with the different capability, high throughput, low throughput, long reads, short reads, but in the same time it's very difficult for the application, for the clinical application to make a selection, and also looking for the less rich countries. I put in this also Italy, for example, now. For the healthcare system it's very difficult to select the instrumentation because we'll change every month, and then we have a new technology. This is one of the problems to move in this path. Looking, this is a very incomplete list of the companies that are already in the market, and also other that are already more or less in the market. There is every week some announcement that there is new technology in this field. This means that it's not an easy area in which we may move with strong support. There is another task that is obviously move from the research to the bench side, this type of technology. There is no doubt that there is a very reduction in the cost. This is a joke. You see that is cheaper than a lemonade squeeze. This means that the cost of the base is decreasing very much, and obviously probably in the future will decrease more and more, and then for sure we'll have this in front of our laboratory. There is no doubt that for the people working in the healthcare system, this new activity is a convergence of chemistry and bioinformatics. This is, you see that also is manually complex. We have to move in automation. We are sufficiently skilled in automation in the healthcare system, but also there is this very large cognitive components that is really a very difficult task to move from this to set up the right pipelines and to give the right information to the patient. I am afraid that in the future, if we don't work in the right way, we'll give a lot of wrong information to our patients. This is my feeling. This is this type of activity is focused on, there is a wet part that is very well known. There is for sure a bioinformatics part that is another part in which also we may do a lot of mistakes, not only in the wet. And there is different parts. We have no time to move on all these parts, in all this part, but obviously there is the image capture, the processing, the sequencer read analysis. There is the variant interpretations. This is another strong task. And you see that many steps that we'll have to fulfill to have the right information to our patients. I would like to outline some of the problem to try to make a parallel between the research and technology and the laboratory medicine. There is no doubt that the next generation sequencing in the routine clinical practice is an opportunity for parallelizing genetic testing, because we have a lot of information regarding the genetic, and then this is we are always in trouble with our patient to give in the right time an answer to the clinical problem. There is also a big challenge. You see that there is a big challenge for storing and assessing complex data. More or less at the end of my presentation, I show you a flow chart from a Canada center that is really outline how we should work in this field in the future. And also is we are working on this, and it is very difficult to identify the clinical relevant and validated genetic variants. I outlined strongly clinically because sometimes, usually in the majority of the cases, we have more than one information for our patients. It is difficult to move in the right way. We have also to maintain the turnaround times. It's very easy to collect the sample of the patients, but we have to maintain short time to give an answer. This is an organizing problem that is not so easy. Sorry, this is too fast. And we have to guarantee the clinical quality and also to make automation. In this part, we have another problem that is very crucial in this part, that is the counseling. Counseling is very important at the beginning, is very important at the end, because is a necessary steps to solve also the data that we obtain from this new technology. And another task that the people living in laboratory medicine knows very well is the integrated all our data with the IT systems in our hospitals, because this is really in the future a collaboration between the laboratory and all the people working in the lab. Which type of diagnostic spectrum we have to do? We are still in our lab, for sure, Sanger, but this is the way. We start now many activity in the multi-gene panels activity, but we're moving more and more in the whole exome on the whole genome. And then at the end in the flowchart, I show you how a whole genome could be useful for solving some problems. One of my, I would like to try one of the points that for sure, this is the reason why we need this technology. There is no doubt that we start for the clinical phenotypes that are very, very heterogeneous, depending of the locus heterogeneity. This means that for many disorders, we have the multiple genes, but there is also a strong allelic heterogeneity with a lot of different mutations. Altogether, this type of variations may in charge of the clinical phenotype. And then at the end of our activity, we may link all this information to the phenotype of the patients. And, but there is also no doubt that this type of comprehensive approach will move from diagnosis to prognosis to treatment to counseling. Outlining the points, I told you that there is the wet part, there is the bioinformatics part, but before and after, there are very strong points. One is the indication for testing, that we have to educate the people to ask, otherwise we do a lot of activity without any solution. Is very important counseling, that also is important at the end. There is a communication of the information. and also to integrate completely this type of activity in the clinical decision making. And for sure, this is not, sometimes in our experiments, in our data, is not so obvious to obtain some results. In the part of the interpretation, I think that is this part, the benign mutations or the clinical relevant mutation, is, I would like to say, is an easy task, but in the middle, for sure, we have still a lot of doubt doing this type of activity. And there is now, we have a lot of support from the bioinformatics, from the databases, but I think that we need some years that we have to collect many information regarding our patients to be more strong in defining the clinical pattern. I would like to outline, being people used to work in laboratory to be sufficiently precise in my activity, that there are many uncertainty in doing this type of activity. There is no, this is a long list, we have no time to be on time, but there is a problem of capture efficiency, there is a problem of sequencing of chemistry errors, there is a problem of the errors in alignment, and so on and so forth. You see that also bioinformatics, but also the interpretation of the pathogenic variant is not very easy. This is one of the problems that I show in front of me. And the key points in this activity is the clinical utility. I mean that we have to always to have in mind that in front of us there is a patient, and this patient asks to us what is really clinically useful for his future. And one of the problems is which type of variant are really clinical actionable. And we need to develop an evidence-based scientific standard to evaluate this data. There is a risk of overinterpretation or unnecessary medical action. And also we have to have a careful selection for patients, genome sequencing and counseling. I think this is only a few points, but are critical for defining the right way to reach the goal to be useful for the patients. This is only a short story. Now I show you very few examples to give you an idea where the laboratory medicine may help in this way. Now we have several new genes that has been detected by next generation sequencing. There are dozens of this type of information. And this is one proposal. This group published a paper that in 50 hours with a short protocol, they may try to define in the neonates some specific clinical patterns. This is useful because in the past usually we had to wait in time. And then this is 3 different examples. We are not to move on, but in these 3 patients, they are able to detect some variations. And this obviously is very important, to start with the right therapy and eventually to start with the right information, counseling to the patient to have another child in a short time. We may also do this type of test for changing the therapy. These are 2 brothers with a specific disorder, dystonia, that is sensitive to levodopa. They are not, the outcome was not so good, but they do the genome sequencing. They detect another mutations in another gene of the metabolic pathway, and then adding the another therapy, the 5-hydroxytryptophan. This is a serotonin precursor. The clinical pattern improved very much. We may improve in the fast diagnosis. We may improve in the treatment. This is the scheme that I tell you. This was recently published by the Sick Children Hospital in Toronto. And I would like to summarize. This is very complicated, not readable, probably slides. But I would like to say that this is the path that we should have in our hospital. This is the patients. The patient reach the cardiologist. This is a young children with the cardiomyopathy. And then when there is the suggestion to move in the genome sequencing, in genome activity, they have to pass to the genetic counselor. This is the part in which this specific professional activity is in charge to explain what it means to do the genome detection, which are the opportunities, which are the pitfalls, which are the risks. And then in this way, also all this information will move also with the phenotype data in this informatics center, which they are connected with many databases. And then after this wet part in which we select some variants, we return, we may have two ways. Information regarding this specific disorder, this is actionable variants, this one, or related to some specific pharmacogenetic test. But there is also other actionable information not related to the specific disorder, because it's not uncommon that during the genome we have what they call the incidental findings. Now the people prefer to call secondary actionable variants, because it's less psychological important for the patient than called the incidental. And this way we have to return to the, for your genetician that may try to evaluate with the counselor which is the importance of these variants. This is, you see, is a work, is a group work in which we have many person working on this. You have in informatics, you have the specific clinicians that are in charge to try to solve the problem. We may do also non-invasive prenatal diagnosis. It has been published that it's possible to have the information regarding the genome of the fetus. There is no doubt that there are already in the markets many companies offering the genetic testing for the aneuploidies, for detecting, for screening aneuploidies during the pregnancy. We may have information, we may detect monogenic disorder by this technology during the fetal life in the maternal circulation. Now plasma is really one of the tasks for this type of the future laboratory medicine activities. There is no doubt that also the microbiology may enjoy of NGS. There is no doubt that the classical microbiology part is moving to the genomic one, typing the genome, or moving in the metagenomics. And there is no doubt that the microbiome is one of the applications that are moving very fast in many laboratories, because there is a lot of publication regarding the importance of this type of test in the world. This is a short scheme in that probably with dietary manipulation, you may modulate the microbiota that may increase the production of short-chain fatty acid, like acetate propionate, that may have a clinical activity, decreasing the appetite, decreasing lipogenesis, inflammation, oxidative stress at the liver level, and also lipogenesis, adipocyte number, increase the gut hormone production, and this is probably will improve the treatment of this patient. This is not only is a single simple scheme, but now there are many recent publications that are linked to this type of opportunities. Cancer, only a few words, because I know that before my presentation there was Lucio Pierpaolo that already did a very excellent presentation, but there is no doubt that also in front of us there is many examples of neoplastic condition which may move in the cancer susceptibilities, in which we for sure BRCA1 or the colon genetic testing that may give us some information regarding this, risk assessment, risk management, somatic mutation, and so on and so forth. And in my opinion also the laboratory should be ready in the future to move in the plasma determination, because also this type of technology is very sensitive, and really we may have information regarding the variation of the different circulating biomarkers during the treatment or during the evolution of the disease. I would like to try to reach the end, to be on time. And this is a recent paper published by Federico in the United States that is a practical scheme how the genome information may be helpful for our patients. And I summarize briefly to you. This is a person that has a mutation in the MLHA1 mutation that we know is a risk for Lynch syndrome. In reality, the patient developed the cancer, the colon cancer, and this person did the genome both in somatic and germline. There was BRAF was wild type, and this is normal for this type of clinical pattern. It was wild type for KRAS, and this way it should be sensitive to cetuximab for the EGFR inhibitor. The clinician decided to give you the CRC treatment with the folinic acid, 5-fluorouracil, irinotecan, to utilize this type of clinical oncology approach. But doing this type of drugs, they have in the genome information regarding UGT1A1 and DPYD, that are two of the genes that are in charge of pharmacogenetics. In this case, we have two mutations that they consider to reduce the dose of irinotecan. There is one mutation also in this gene and also reduce the 5-fluorouracil. But in the same time, this patient has a cancer, but is not. The cancer is a risk for adverse reaction for thromboembolism or for venous thromboembolism. And you have information regarding the cytochrome 2C9 and 2V-cork for driving the therapy of for the thromboembolism. And there are, you see, some mutation, and they decided to start with the lower dose to avoid the bleeding. But being the cancer, being the also thromboembolism, we have also some depressive situation, and you have to take antidepressant. But this is the real life in which the patient has to approach on this. And then you study the cytochromes. There are two variants, and then they decide to reduce the dose. This is only a practical schema in which way this type of information may reach the laboratory. Challenges. I finished in the last 5 minutes. This is the opportunities. Now I would like to transfer also the pitfalls of this type of activity. Being people working in laboratory, we need to have a common standard for ensuring the reliability of MGS results that up to now does not exist. There is no any protocols that is really very strong that could be utilized in many laboratories. For sure, there are some single laboratory that works very well, this is no doubt. But being in the healthcare system, this is the goal. We have to apply regulatory requirements. How we validate or we establish a verification of performance specification. Our lab are certified, are accredited. In which way we may do this type of activity? We have to set up quality control procedures. Quality control should be for the wet part, for the NGS, but should be also in bioinformatics, quality control. And we have to check all the different points. We have to set up an independent assessment of their performance, proficiency testing, alternate assessments. There are no doubt that in the United States there is a strong effort to do this. You know that already it's difficult to have good proficiency testing for many genetic disorders. Probably the way is this to an evaluation of the technology, not the single application like the classical Sanger sequencing. But for sure to have this proficiency testing that may declare that you are working the right way is an absolute need for this type of activity. And the other problem is to have the reference material, in which way we may validate our test. If we move in a new test, for sure, this is the time. Okay, I finished. There is the problem to have the right informed consent, to outline the ethical considerations. But I spoken on genome. There is no doubt that the family is much larger. Probably in the future will have more holistic vision of all the different technology. I am at the end. And I say that we are really in the time to move from the bench to the bedside. It's not an easy task. I wouldn't like to have this small van going around with for the DNA testing, because it's the risk for the patients. And hoping to not boring you too much, I would like to thanks, but my thanks are really to all of you for the patient to hear me, but particularly to Franco, that is really a great person for Italy, for Naples, for international level. It is a friend. Thank you very much.
Maurizio, is there any question or comments? If not, together with my co-chairman Francisco Ramirez, we thank the 4 speakers of today's session and all the participants. Thank you very much.